Okay, so good afternoon to the second part of this day where we are actually doing the annotation. My name is Matthias and I, together with Kadir and Markus, created this annotation platform. And I will talk now for 30 to 45 minutes about how to use it in detail, how to create the annotations, what we are asking you for, and afterwards at 2 we will start the actual annotation upload, where we will ask you to state questions in the chat, and if there's a need for a call in, please mention that in the chat as well, and we will contact you and give you feedback in real time. So the platform is situated at DANIO-CODE.cvin.org. So here you have the two basic interaction modes, is to add an annotation and basic views and annotation. You can add annotations in two ways, or with using two different interfaces. One is using a web interface, and the other one is you create the CSV file and then upload this on the site. I will start with the web interface, and since it has the sections in smaller chunks, and later on explain the CSV upload, which again, both of them do the same thing, just in different means. So if you go to the web interface, as Carsten said in the morning, we have a top-down approach. We start with the series and then go to bar samples, assay, applied assay, sequencing, and then to the actual data annotation. And for the series, for all of these sections, we have required and optional annotations. The required fields in the web show up as, if you don't enter them, we have a form control for that. So you are reminded to please enter that data. And on the example XLSX file, which is in the help section here, or at batch upload, you see it as a green column color. What are the required fields? And we also have optionally required fields, which means these fields are only specific to, for example, if you have a sample type tissue, then you also have to specify what anatomical term corresponds to this tissue. But back to the series annotation. So for the series part, we need a title. And in order to shorten this, I will now create just a dummy upload, so no biological interesting things happening here. The required fields and sections are title. Then if it's public or not, this is currently not so not so sensitive, because all the data we are collecting now is public, but in future it will just say if public, then it's to general audience, and if it's not public, it will only be available to the DANIO-CODE consortium. The next is then the two series types. If it's a survey or a case control, the difference is simply that for case control, you specify biological controls. For survey, there are no biological controls. Technical controls are still possible. And so let's start with case control again. The thing is similar, it's just an added layer on top. So how to enter the bar sample and technical replicates. You enter here the number of technical replicates per bar sample. So if I type the vector 532, this will correspond to 3 bar sample groups, where the first one has 5 biological replicates, the second one has 3, and the third one has 2 replicates. You will later on see how this is then visualized. And I could also specify the maximum number of replicates if I have a larger number of bar samples, then I don't enter a vector. I just enter, for example, 5. This will create 5 bar samples. but I would need to state how many biological replicates are there per bar sample. But let's continue with the vector. And the next required field is the number of different assays used in the series. I will go here for 2, and later on we will specify which of these which assays were used specifically. And since it's a case control, we also have to enter the same like with number of bar samples, how many bar samples, how many controls with biological replicates we have. So let's enter two controls with 2 and 3 biological replicates each. And also a description is needed. This corresponds to something like the abstract or a short overview of the series itself. Again, just some dummy data. And the non-required fields in these sections are published in, where you can specify either a citation or a PubMed ID. And if the data is also available at GEO, you can also sign as GEO series ID here. So if I now go to the bar sample section, sorry, you see we have now the setup we described before. We have 2 controls where the first one has 2 biological replicates and the second one is 3, and 3 non-controls where the first one has 5, the second one is 3, and the third one has 2 biological replicates. We can still alter this, and this step, in the later steps, we can't alter the number of biological replicates. So, how to annotate for bar samples? Here you can select either using the selection rectangle or using the button where you select all, since probably many of the annotations are the same for all of the bar samples. There may be smaller differences between the groups, but in that case you can annotate a large number of bar samples in one step. So the required fields here are lab, which is the lab which handled the bar sample, then and can be all the labs have to be, before doing the annotation, have to be added using the link up there at lab, where you just enter a lab name, and then it will be available on the website as well. The same goes for the CSV upload.
Okay. So maybe it's better that you will re-explain after for us, so you make the presentation for the guys, because everybody is looking here at the screen, and that's a problem.
Okay.
Too much. Is it possible to display on the screen?
No, because then you can't capture it, right? In the mirror, I get a really small screen, and then I need a large screen here. That's the problem.
But it's better. Can you do?
Yeah, I can do it twice. Yeah.
Twice. So please switch off the.
Just listen to me. You can still go to the homepage and follow there. Yeah, sorry for that. We just some technical difficulties in the room. So back to the bar sample. As I said, first required field is lab, and we have already here a list of where some of you already entered their labs. Also, this lab also corresponds to sequencing centers, so it's not only labs, because you also have to specify later in the sequencing section which lab or sequencing center performed the sequencing. So let's go for Fiona's lab. The next required field is the genetic background of the fish. This is the common list of names. If it's not known, please enter the wild type indicator down there. Maybe some short comments about the controlled vocabulary. This is mainly based on, if it's zebrafish related, this is based on annotation on the ontology provided by CFIN. So with that we hope to cover all the important bits. After the genetic background, we have to specify which kind of biosample we used. Was it a whole organism? Was it just a tissue, stem cells, or cell line? And as I said before, for whole organism and as for tissue, we get new fields added where we have to also define the developmental stage and the anatomical term. These are required. Optionally, we can also say the time post-fertilization. This for a whole organism, of course, there's no anatomical term, but there is still a developmental stage, which is a required term here. And since it's a case control, we also need to state which are controlled for. So let's say the first two are controlled by the first one here, by the first control sample here, and the last one is controlled by the second one. Maybe a short comment about the interface here. If you haven't selected anything here, any samples or any of these title cards, you can't select anything on the right side. It won't accept anything. Just if you run into that error, that you know, okay, you have to select first a field, and then it will be applied on it. So if we have entered everything, and each biosample group has also a description field where you can enter some specific terms if you want to describe it further. We also have 3 more common fields, which is a source field for the source of the zebrafish, if it's was from a zebrafish distribution center, or from which distribution center, or maybe, yeah, wherever, if it's available, this information. These are optional. Treatment is also an optional field. Here you can say like heat shock or different kind of treatment applied to the zebrafish. And in the last field here is mutation description, where you can say like SOX32 knockdown. And with that we have all the fields. Maybe I should just show you that it is really applied to all the fields. And after biosample we have to define which, or declare which assays we used. We can therefore either use already already used assays, or we can create new ones. And the required fields here are all you can see. So you have to state a lab which performed the assay. And maybe some short comments about what we understand under assay. For us, an assay is a procedure applied to a biological replicate to generate a sequencing library. And so we need, this is done by a lab, which I have to declare. Then there is a type, and here we have hopefully all common assay types which occur in our database there. If not, please contact us and we will add them immediately. And so there are, of course, also different types. For example, if I use any IP assay, I also have to declare a target and a control target. So we have here like you state any transcription factor, for example, and the control target can be whatever is your control. You can also, if it's an input, just state input. If it's a mock, say mock. And also a description, which can be the short version of the protocol used, short specification of the protocol or kit used to prepare the essay. And since we stated at the beginning in the series that there are 2 essays applied in this series, we have now to fill out two different kind of essays, and I will add, so you can see you can also reuse already established ones, and I will have one RNA seq and one chip seq as essay. And afterwards we actually assign each biological replicate the essay to it, which is basically the creation of the sequencing library. So again, here you can select all replicates. Keep in mind we are now on a biological replicate level, therefore you don't select the whole bar sample, but you can select subgroups of biological replicates. But for the sake of time and ease, I'll just select all of them. And so we have here RNA seq, and you see that it is applied to the to all biological replicates, and we also can state now the technical replicates for each applied assay. And if we add chip, you can also see that the control target is also available as an assay. It has the keyword control in it, and later on, if you now hit enter sequencing, these biological replicates will be linked. So if they have the chip assay and its control assigned, then it will later on, we know in the database that okay, this has a control in that applied assay. So after the applied assay, we have to specify what kind of sequencing was done. Here now you can select the technical replicates based on the assay used, or again select simply all of them, and state which sequencing platform was used, which lab or sequencing center did the sequencing, what kind of instrument was used to actually do the sequencing. If it was single-end or paired-end sequencing, this has effects later on, because for single-end sequencing, we only accept one data file per sequencing, and for paired-end, we expect exactly two data sets. And since we also have an RNA-based assay in our series here, we also have to declare a strand mode, which if you have no RNA-based assay in your selection, this field does not appear. So don't be confused if it's not there. It's there if it's required. If it's not required, it's not there. And we also have to state a date. There, please use an as accurate date as possible. If you only know the month, just say the 1st of the month. If you only know the year, it's the 1st of January of this year. So you can select here the years. And yeah, so these are the required fields here, all there on top. So it's the platform, the lab, the instrument, if it's single or paired end, and for RNA-based assays, if it's which kind of strand mode was used, and the sequencing date. Optionally, we can also specify what was the max read length and what is the chemistry version. used to prepare the specific sequencing. And last but not least, we now have to assign the file names to the technical replicates, and here you will use the file names you used when you sent the data to CFIN. You have to specify it as I'll show you shortly. So it's your folder name of the lab / then the exact location of the file. So if you have like subfolders, you add them, you mention them here as well. And if you're not sure how your files are called or how your lab directory is called, you can view them in a few file names up here. So if we now again, we selected single and sequencing, so we only have to declare one file per technical replicate. I'll just go quickly through it. Of course, normally you don't have the same data set for all technical replicates, but for the sake of time, I'll just copy paste the same file path to all of them. Okay, so now all the technical replicates have a data file path assigned. If I now hit submit, I get, after a short while, since there is some processing happening in the background for larger files, it can be a little longer, but you will then finally see a successful upload annotations notification, where you can also see the details, while you see in a concise view what we just did. And so this is the data upload using the web interface. There's also a CSV upload. So you can enter it using the tab up here with Add Annotation CSV Upload, or go back to the main page and enter and go there to the CSV Upload. Here we have everything again listed. What are the required fields? How should they be? How should they look like? Because for the CSV, they have to have a specific format. I'll show you shortly. So, but again, all the terms are the same as before, so they should be familiar. It's just a different kind of format. So this is now the sample batch upload file, which is also available here in the help page and in the batch upload page. And this is how the CSV file has to look like in order to be accepted. The first line is just table headers, and these are the names of the different annotation terms. So I'll just format it quickly. And so the one little different thing than in the web interface is that these columns have the section name before each column name. So for the series section, you always have series and then double underscore. The double underscore is important because we pass for that. And then it's just like just the different names, but you can reuse simply the sample batch uploads with your specific data, and then it should be fine. But just to keep in mind, there should be always the section name with a double underscore in the header file, in the header line. So if we go through it again, you always have an internal ID, which is the ID internal for the CSV file. In this setup, we have one bar sample with where we do one chipset experiment with 3 different targets. One of it is a mock. So we have 3 lines because we get 3 sequencing files. Actually, it's double and paired end sequencing, so we get 6 files in total, but you sequenced 3, you sequenced 3 samples, so 3 lines, and since the series is always the same, it has the internal ID 1. If you want to reference already assigned an object in our database, you can use the specific ID here. You can see the IDs in the view annotation section. Afterwards, it's the entrances are as before. We have a series title, a type, if it's public, if it's the description of it, the publication, and the SR or GEO ID. So we don't have to declare these for all 3 lines because all 3 lines have the same input. So we will copy them internally. You don't have to copy paste anything. If they are different, you have to declare that the new thing. But if the line below has the same thing in the cell, then the cell above, it will be just copied, so you can leave it empty. So after the series, we have again the bar sample. With again, we only have one bar sample, so there's only one internal ID. And again, we can declare, like if it's already in a database, we can specify it there. And then we have the same terms as before. Just one small thing here is bar sample organism. It is a field in our database, therefore you have to put it in your CSV, but you can just leave it empty. It will be automatically assigned as DANIO-CODE, since we are currently only using DANIO-CODE. But as stated before, all the terms and names are the same as before. Please use controlled vocabulary terms here. We check just for these terms. You can find all the controlled vocabulary terms in the help page. I'll quickly go to it. Here in the bottom you have the table of all the controlled vocabulary terms. So if you're not using these terms, you will get an error message later on where we will state that you used a term different than we are expecting. So let's go back to the CSV file. Again, maybe mentioned the CSV file can be simply created using Excel. You just save it as a CSV, and then you can simply upload it. So okay, we have bar sample. After bar sample, we have the bar sample replicate. These you didn't have to specify in the web interface, but here you have to specify them. But we only have one bar sample, so and with only one replicate, so the bar sample itself. So we also have here just once. But now in the assay part, in the assay section, we have something new. Now we have 3 different internal IDs because we have 3 different targets we have 3 different assays. We have the chipsec for with the assay target mock, 1 and 2 for histone modification, methylation status, and so therefore afterwards we have also 3 different kinds of applied assays, but there is nothing really to state here since the connection is already made, because a bar sample, a biological replicate, and its assay have to be on the same line. Therefore, we know which assay was applied to the biological replicate. But applied assays can have controls, as we saw before in the web interface, and you simply state that by using the internal ID of the control. So applied assay 2 and 3 are controlled by the applied assay with internal ID 1. After the applied assay, we have to state the technical replicates and then the sequencing, but here it looks similar to the things before, so I don't go in detail here. Just now we are at the in the data section, where I can either specify a URL path to the file or the local path on our CVIN server, which, as mentioned before, you can find in the View File Names link up here. So the file name, as before, has to be parent folder name, so name of your lab folder, and then the exact destination to it. And if you look, we have paired end sequencing selected, so we have two different files here, and yeah, that's one. So therefore, one line is the whole thing we went through before for one data file, and again, it will come to the same result. So if I go now to the batch upload annotation, CSV upload, you have here a long list of all the different explanations of the terms, and if they're optional or required, because as in the web interface, the same terms are required for the CSV file. Then I simply choose my batch upload and hit submit. Then I can review everything before. So now it's in a limbo state. If you see that something was misassigned, because it can sometimes be confusing with the different lines, and in CSV in an Excel sheet we show it here in a more organized way in all the sections as we have in the web interface, and so you can control easily. And if everything is correct and we can't find any mistakes, we simply hit commit here, and yeah, it is committed, and we can view the annotations. You see here we have the series here from we just uploaded with the CSV file, and below we have the one uploaded using the web interface. And here you can view then all the bar samples, the assays used, the applied assays, the sequencing, and the data. So that's it, how to annotate things. Again, look at the help page and the wiki, where we have the tutorial written again, and the help page we have all the terms listed again, since terms are often overloaded with different meanings. Here we try to be as explicit as possible about the different terms. With that, that's it from the demo here, and we can start using, or you can start doing the annotations on your place, and we will be contactable in the Skype chat. So see you there, and good luck with the annotations. Thanks.
You want to address the questions first?
Sorry, yeah, we can already address questions.
So there is a question of maximum file sample number.
No, I don't need it.
I said there is no maximum.
Yeah, there's okay. So there's a question if there's a maximum number of bar samples. There's no maximum number of bar samples. And there is another question where they. Okay, yeah. So please enter it using the vector format as before. So don't say we have 10 bar samples, state how many replicates you have of these 10 bar samples. So it would be like, I see the example here, 10 bar sample with a maximum number of replicates. So please enter the number 6 10 times. I'm sorry, that's cumbersome. I can't currently fix that problem. I think I know the solution for the problem, but I can't fix it. You can do it using the vector format. This works, and then you don't have to state the maximum number of replicates. Yeah. No. Any more questions? Okay.
Yep, there's one.
Yes, Katia is showing it to me. And bar sample. You should be able to delete them using the X button on top. There is a limited way how to go back and correct the previous entry. The problem here is that since these terms are deeply connected, it is quite difficult to say, for example, if you want to change the number of bar samples you have in general, we don't know then, is this then a new control or not? And therefore our current solution there is to ask you to restart it. We tried to make the web interface as fast as possible to enter data. Therefore, we think that's a better solution than the other way, because with the other way, we can't be sure that we get all the correct annotations. Okay, so there's a question about the hierarchy between bar sample, assay, and sequencing. I'll just read the question. I have one biological sample and then sequence it. Later decide to resequence it to get higher coverage. So this is then an example of a technical replicate. You simply state it as two, in the applied assay section, you state it as two technical replicates have then two different technical replicates to assign sequencing to. Yes, so in the CSV it has the same bar sample, internal ID number, the same applied assay number, but a different technical replicate ID, internal ID number, and therefore also then a different different sequencing internal ID number. Concerning if each line should correspond to the SRA, it is sufficient if each line, if the SRA corresponds to the series ID and not the actual sample ID in the SRA. Okay, are there any more questions? Yes, please. Each lab has to add their own labs, since there are so many different labs inside one series. we can't capture them all, so we're asking you to first provide them using the add lab link on the page. Okay, so any new questions? Maybe shortly, you can actually modify and delete the series, but only concerning like if you have a different publication. We don't want to do it with the later steps again, because these are two numbers effects which have it later on, since all of the strings are then connected, and if you change one thread in the middle, we would have then to always recheck everything, and we can't do that at the moment. No, the sequence, so the question is, I should put technical, it goes back to the question when you have two technical replicates, how it looks like in the CSV file. So it is then you have, once you have two different internal IDs, all the other ones also have different internal IDs. So the internal ID for the technical replicate is 1, 2 if you have two technical replicates, and therefore also the sequencing internal ID has to be 1, 2, since it's a one-to-one relationship there. Yeah, I can't edit the say. No, it shouldn't be a browser problem. The edit essay, wait, okay, so the question is, can it, I cannot edit the essay. Then, like, so I'm guessing they are in the essay detail view. I just have to go there. And yeah, so yeah, then you please create a new essay and reassign it there, because or wait a moment, it's there should be an edit button. It's currently not visible. I'm sorry for that. Shit. If you have a spelling mistake in the lab, simply create the lab again, and we will fix it on our side. The thing here is we want to leave a little more control, because there are different, yeah, with all the different nationalities and languages, it's often difficult for us to make a strict control on it, of the format. So yeah, if you simply re-edit it, it's fine. Any more questions? Yeah, so my email is on the wiki, otherwise it's also in the help page, I think. So it's matthias.hörtenhuber@ki.se, or just simply we can post it in this Skype chat as well, and I'm open for questions and suggestions there. So then. Yeah, so if you select a sample as Polé or Rivasira, you can put that into the description of the bar sample itself, since it's not specifically or not traditionally a treatment. But for the assay, edit assay thing, I can fix that tonight. So if you hang with us, you don't have to recreate everything. You will be able to edit the assay, not how it was assigned, but like if you made if you put the wrong description there, you can't change the assignment. So linking to other databases, it should be the SRS ID. And yeah, and you can put the data into the best way would be in the series description there. Okay, there's a question concerning so yeah, maybe a short comment concerning browsers. For the web interface, all modern browsers are supported there. So everything like for Internet Explorer, it has to be Internet Explorer 10 up. Just check the question there. So maybe also a short word about the view. If you finish the annotations, currently it's simply a table, but we will expand this to more sortable tables and views, including biomat-like data export. Okay, last question. We have an internal sample number. Is the question, would you like to keep it somewhere to be able to keep track? Where can I put it? If it's for biosamples, you can put it into the biosample description. Okay, so we will stop the stream now, but keep the Skype chat open, and yeah, we'll answer your questions there. If we can't answer them in the text, again, contact us and we will call you. With that, thank you and have a nice afternoon. And